Hi, this is Dr. Dan Guerra coming to you from Vera Med Studios in combination with Authentic Biochemistry, my podcast. Today is the 14th of September, and we're going to be discussing a particular adipokine, which I will define in a moment, that regulates not only lipid metabolism from the adipose, but also a host of other normal physiological and pathophysiological conditions, depending on whether or not the human body is in some kind of stress, and in particular obesity or other kind of process that can induce dyslipidemia. So an adipokine is basically a proactive, secreted hormone type compound that acts as a cytokine, but it comes from the adipose tissue. So typically these are glycoproteins that are secreted through the ER and Golgi apparatus across the plasma membrane of the cells that secrete them. And when this occurs, the adipokine then functions to signal to various other tissues. And the adipokine itself is generated because of signaling that's caused homeostatically throughout the body. So with all that kind of prolegomena, I'm going to start this PowerPoint here. All right, so let's get started here. Slideshow. All right, so we're going to talk about lipid biochemistry and physiology, and I'm going to be using primarily a review article that was just published in 2019 this summer. We are also going to be looking at a couple of other papers where I've got some of the figures for this talk. And then after this talk, I'm going to go ahead and use this review of leptin to look at some specific new research papers, some fundamental basic research papers about leptin when you have this background. Again, I'm Dr. Daniel J. Guerra, and Vera Med and Authentic Biochemistry is where I'm publishing from this particular video lecture on YouTube. There's my email address. You can contact me there at any time you want and ask me about the talks that I give or about my companies, Vera Med and Authentic Biochemistry, and we can help you take a look at the basic scientific research that's published in refereed journals and guide you through the use of that information, vetting that data and looking at the evidence and verifying the evidence against the largesse of the locus of all the other published research in that area. We work with pharmacists, we work with clinicians, we work with nurses, we work with nurse practitioners, all aspects of physicians, but we also work with graduate students, medical students, veterinary science students, pharmacy students, and indeed lay people who are interested in knowing more about basic research, and anyone else who has an interest in basic molecular biosciences. That's what I do. My PhD is in biochemistry, and what I do is normally teach biochemistry, graduate biochemistry. All right, so let's just get going here. So what are adipokines? Well, they're signaling molecules, and they're produced primarily from the adipocyte. So adipocytes not only produce these proteins called adipokines, and amongst the subset of them, types of them, or specific names of them are adiponectin, leptin, which is going to be the discussion today, visfatin, and resistin. Now, each of these adipokines have different physiological roles. What kind of roles do adipokines have? They have a role in obesity. They have a role in arthritis, both rheumatoid arthritis and osteoarthritis. So there's a pathology there. They're involved in cardiovascular disease, obviously a pathological condition that's basically number one in causing morbidity and mortality in humans, and even insulin sensitivity. So leptin actually helps control insulin resistance. In fact, there is such a thing as leptin resistance. So besides adipokines being generated by the adipocyte, which is all in the visceral depot fat, you also get complement produced by adipocytes. You get regular cytokines, which can be pro or anti-inflammatory. And you have chemokines, which allow for cellular trafficking towards site of, for example, inflammation, right? So that comes from the International Journal of Molecular Sciences, published just very recently, you can see. That's an open access publication. This also is an open access slide that I picked up. This gives you a little bit of idea why people are interested in adipokines. So in obesity, in an obesogenic state, you've got free fatty acids increasing, you've got leptin increasing, tumor necrosis, that's an adipokine, tumor necrosis factor alpha, which is actually a pro-inflammatory cytokine, IL-6, pro-inflammatory cytokine, and MCP1, which regulates all the trafficking of these cytokines. Those all go up in obesity, whereas adiponectin goes down and LPL goes down. So what happens during obesity is you get less lipoprotein lipase, less adiponectin, but an increase in a lot of these pro-inflammatory cytokines, increase in leptin. You're going to learn today what an increase or decrease in leptin can do. But all of this can cause, in type 2 diabetes, for example, insulin resistance. Now, in centenarians, very old people, over 100, what do we find? We find a decrease in free fatty acids. We see a decrease in leptin, but we see an increase in adiponectin and an increase in lipoprotein lipase. Now, that's interesting because people who live to be 100 tend to have lower levels of circulating leptin and free fatty acid, higher levels of adiponectin, which kind of works as a contrast or contrary or even contradictory to leptin, and then lipoprotein lipase, which basically is an enzyme that regulates removal of fatty acids from circulating lipoproteins, allowing them to be taken up by cells, removing them from circulation. Now, when you have lipid dystrophy, which means there's some kind of problem with lipid metabolism, such as ectopic lipid deposition, by that I would mean, for example, around cardiovascular system or in endothelial cells, that's causing the clogging of arteries, atherosclerosis. You also see a decrease in adiponectin, right? And there you see an increase in centenarians. You see in lipodystrophy, you see a decrease in adiponectin, but you also see a decrease in leptin. But you get a huge increase in lipid deposition, and you get a huge increase in free fatty acids in circulation. That's insulin resistance. This is insulin sensitivity when you have this kind of biochemical molecular phenotype, and this is insulin resistance. So you get an idea, this is just basic generalization of what you see with adipokines. All right. So more now about the specific adipokines we're going to talk about today. Here's leptin, okay? Leptin helps regulate the synthesis of thyroid hormones. It decreases glucose-stimulated insulin secretion. This is generally considered a good thing because excessive insulin secretion can lead to hyperinsulinemia, which can lead to insulin resistance. Leptin also increases heart rate. It also increases blood pressure if it's in high circulation. It can activate various kinds of immune cells, which is what we're going to talk about. It also regulates appetite. That's the general feature of leptin. So when you have a good store of leptin, what it does is signal to the brain, particularly to the nuclei of the brain that control appetite. And it controls appetite, regulates appetite. It controls metabolism via the central nervous system, and therefore energy homeostasis by regulating the kind of biofuel, for example, glucose versus fatty acid oxidation. Leptin also regulates the menstrual cycle in mammals, and it regulates solid bone mass and mineralization. So it's a very, very complicated and highly regulated and also potent adipokine, leptin. Now, let's take another look at now leptin in the specific. Okay. In normal leptin, the brain acts on the signal from a normal fat mass. You have a normal appetite, it's regulated. You have an increase in metabolism when leptin is synthesized, generated, and sent to the brain. And your weight is basically normal, right? This is when you have homeostatic state where you're not too thin and not obese. Now with low leptin, the brain acts on the signal, okay? So that means there's a reduced fat mass, you get a reduced leptin signal. That's what happens in a low leptin. And what is the response physiologically? The body is in a starvation mode. Why is it a starvation mode? Because if you have a highly reduced fat mass and therefore less leptin being synthesized and secreted and making it to the central nervous system, It's telling the body that you don't have any storage depot fat. That's a starvation mode, okay? So what that does in starvation mode is it turns on the appetitive system, so you increase the appetite, and it decreases metabolism, right? You're going to suppress metabolism because you're not going to want to burn all those calories because you don't have much in reserve. The third type, and this is again a pathophysiology, is leptin resistance. The brain only receives part of the signaling because the leptin is no longer as potent of a signaling molecule because it doesn't function as well at binding to its receptor for various reasons we'll get into. And that induced, you get hyperleptinemia because you have excessive fat mass. So you have a lot of leptin produced, and just like if you have a lot of insulin produced or any hormone, what happens basically is you start to get resistance in the periphery. That is, the signaling starts to be tamped down because there's too much of the compound that's inducing the signaling via ligand binding to its receptor. So what does this do when you get leptin resistance? Well, these aren't good these are pathological states. You get more appetite, even though you have an excessive fat mass. So think of this as an obese person. You have decreased metabolism, so you're not burning all of that fat, and so you gain weight. You get an increased risk of metabolic syndrome, which has to do with hypertension and various kinds of mistakes in metabolism, and of course type 2 diabetes, T2D. So leptin resistance, a bad thing. Low leptin just basically means starvation mode. I mean, you're fasting, you get low leptin, especially if you have a lot of lean body to fat mass. You're going to be producing not very much leptin, and that's going to put you in this mode here. Normal leptin, you can see that's maintaining weight homeostasis, appetite homeostasis, metabolic homeostasis. All right. So here's the paper we're going to be examining primarily. Again, it's a review article. I'm doing the review article for today because then I want to go into the literature, dig into the literature, and get you primary published papers now doing active research on the area. So you can see it was published in Nature Metabolism just recently. In fact, the very first volume of this journal just now came out this summer. So what do we learn from the introduction of this paper? They start telling you some basic features to understand what leptin does. To tell you, of course, that the second law of thermodynamics states that energy within a system has to be conserved, and that any change must result, from either a change in the energy input or output from that system. Okay, so that's how a closed system will function in terms of the second law. Thus, among mammals, in which energy is stored primarily as triacylglycerol, which gives you, again, 9 kilocalories per gram. So it's the highest bioenergetic nutritive component that you can store. That's why we are we are organisms which store, we're oleaginous organisms. We store fat because fat requires no water for storage, unlike glycogen, which is a homopolymer of glucose. So we store fat against the concentration gradient. We put it throughout our viscera. And we do that because fat is a very good biomolecule to generate a lot of energy when needed. So remember, the human body evolved as a hunter-gatherer. So hunter-gatherers ate episodically. And so they gorged on a lot of food. We didn't have refrigeration before we learned how to do that. Even before fire, for example, or controlling fire, we couldn't heat the food. So it was a massive ingestion of food. And so the body was regulated in such a way that it turned a lot of that into depot fat via the synthesis of fatty acids, the synthesis of triacylglycerol, and then the entire metabolic sequelae that's associated with lipoproteins in the serum. And then the movement of those lipoproteins to various peripheral systems and organs to deliver that fat so they could be stored, like I said, in the viscera in mammals, right? So that's a very important understanding that we are oleaginous organisms. So any kind of defect in metabolism or pathology that's associated with the major diseases, such as cardiovascular heart disease, such as cancer, or any kind of metabolic disorder, all of those diseases are going to impact or be impacted by dysregulation of lipid metabolism, because again, our fundamental storage and utilization for biofuel and signaling are lipids, not carbohydrates, not amino acids, and not nucleotides. All of those other molecules play roles, but they are not the major storage compound. nor are they because most of those other compounds I just mentioned are water soluble, so they can circulate more or less freely in the serum, although sometimes, of course, they're bound to protein. Lipids, on the other hand, require to be associated with proteins, but usually non-covalently, as in triacylglycerol, cholesterol esters, phospholipids, sphingolipids associated with different lipoprotein classes. Okay, so we know about that. We know that there is any change in weight or fat mass must necessarily be a result of changes in energy input. That's going to be food intake, right? Or energy expenditure. For weight to remain stable in a living organism, okay, energy input thus must be precisely balanced against energy output over long periods of time. So this is just following the second law of thermodynamics, and all living systems seem to do that with varying degrees. And then from one end to the other of the spectrum is where you get pathology. So when food is readily available, adult humans maintain a remarkably stable weight while consuming approximately 1 million kilocalories each year. Collectively, these findings that you're hearing about here have long suggested the existence of a biological mechanism indexing or surveilling food intake to energy expenditure, following the second law of thermodynamics, to maintain the stability of weight or mass of the body and adipose tissue mass. Maintaining the stability of energy stored in adipose tissue, then, is also essential for survival, because lower levels of body fat have, of course, deleterious consequences because it increases the risks of starvation, which, of course, can kill you. And that happens during periods of food insecurity or famine, or again, when you're thinking about the early stages of human evolution, it would be a very common occurrence to have long periods of fasting, high intensity caloric intake, and then fasting can sometimes be days to even weeks, depending on when the next food source with high caloric density can be found, and also with the appropriate amino acids and essential fatty acids and essential vitamins and other nutrients that your body doesn't synthesize directly sensu stricto. So this controls, of course, fertility, the immune response, either an increase or a hyperimmune response or a suppression of immune response, neither of which is a good thing. And a higher adipose tissue mass increases the risk, of course, of predation because you don't move, right? So all of this is tightly regulated, as you might guess. It has thus been postulated, according to the introduction of this paper, that nutritional state, probably from a fat-derived signal, is sensed to maintain the homeostasis of adipose. Of course, that makes sense. However, the identification of that molecule or any kind of molecular component thereof of the putative system was pretty elusive for quite a while. For a while, people thought lipoproteins were the answer, or what was in lipoproteins, such as circulating fatty acid, circulating triacylglycerol, circulating complex lipids. But it didn't appear to be that those were the major regulators of controlling this lipid homeostasis, right, via a rheostat that's regulated by something probably coming from depot fat. Owing in part to the intrinsic difficulty in applying biochemical methods, it's difficult to be a lipid biochemist. I happen to be one, and because lipids are not water soluble, everything you do in the laboratory is increased at least one order of magnitude in more complexity, because you have to deal either with organic solvents to extract lipids and then isolate them on chromatography, for example. Even HPLC gradients require organic solvents like chloroform or acetone, acetonitrile, in order just to separate lipids and determine what's there. And even the extraction, of course, usually requires chloroform methanol or butanol or lots of different organic solvents, even petroleum ether. And that means that you have to have a totally different type of laboratory, biochemistry laboratory. So, and there aren't many people that are lipid biochemists. Maybe that's why people don't like working with organic solvents. They're dangerous, they're explosive, they're inflammable, and they're particularly toxic. So you have to be in a fume hood half of your life when you're a lipid biochemist. I can tell you from experience that is absolutely the truth. So anyway, that was why it was difficult to find these things, besides the fact that these adipokines aren't themselves actually lipids, they're protein molecules, right? So to determine what is physiologically regulating food intake and energy expenditure in vivo, ultimately these had to be discovered. The difficulty was further compounded by the ability of many factors to decrease food intake, both, for example, the appetite control, right? not specifically eliciting an aversive or non-physiological response, including things like nausea and other unpleasant sensations. All right. So more now about leptin and leptin binding to its leptin receptor. Leptin binding to its receptor controls hormones from the central nervous system, food intake, and energy expenditure. As we said, those are central actions. And then in the immune system, a whole host of complicated and interwoven axial interactions with all of the innate and acquired immune cells, humoral and cell-based. So cytokine induction, chemotaxis towards sites of inflammation, natural killer cytotoxicity is controlled by leptin, macrophage activation from moving from the M2 quiescent to the M1 active, dendritic cell activation, very important for antigen-presenting cells into the T cell population, generated originally from the thymus, but found, of course, in medulla and other regions of lymphatic tissue. Th1 stimulation, Th2 inhibition. Th1 stimulation can give you high levels of inflammation. Th2 inhibition can sometimes dampen down the autoimmune response. And of course, naive T cell proliferation, which means it can go to Th1, Th2, Tregs, and also killer T lymphocytes. So all four classes of T cells, starting from CD4, CD8 lineages, can also be differentiated in those subclasses. And the Tregs, of course, suppress, generally speaking, the immune response, something we talked about at great length just last several episodes in Authentic Biochemistry and also here in the video lecture. Other targets for leptin receptor: kidney, bowel, pancreas, and muscle. Reproduction, as I've been suggesting to you, is also regulated by leptin levels. Placental function, trophoblast invasion, fetal function, gonadotrophins and gonadal hormones all are regulated by or respond to leptin binding to its receptor. There's vascular function, as we've already alluded to, atherosclerosis because of the mobilization of lipids and the incorrect or inappropriate deposition into, say, endothelial systems. Angiogenesis, the laying down of new blood vessels, which can lead to, of course, the potential to feed tumors. Oxidative stress, of course, the production of reactive oxygen, reactive nitrogen, particularly reactive oxygen, can lead to mutations in DNA, or oxidation or auto-oxidation of lipids with any level of polyunsaturation. And all of that can lead to mutations, which can lead to oncogenesis. And then even in bone and cartilage, bone mass, chondrocytes, rheumatoid arthritis, osteoarthritis, the same thing. We mentioned already, leptin has a mediating control over all those pathologies. All right. Now here's some specifics. Here's leptin binding to its receptor, and here are part of the signal transduction cascades, which are turned on once leptin binds. These are various tyrosine residues. This is all part of the leptin receptor, which is a dimer. So tyrosine 974, these are canonical features of leptin receptors. Tyrosine 985, 1077, and 1138, all those become phosphorylated by this JAK2 system in cooperation with the SOX3 protein. So leptin binds with the receptor, the binding receptor to the long form of the receptor. This is the long form. Okay, so the long form, there's the monomer. Short form, there's the monomer. So there's a long form and a short form. Here's the dimer, fully composite and composed so it can function as a full-blown leptin receptor. Binding to the long form is dimerization, prompts Janus 2 kinase, it's JAK kinase. Autophosphorylation, which phosphorylates cytoplasmic domain of the leptin receptor. Those are all those tyrosine residues there I just mentioned. Each one functions as docking sites for different cytoplasmic factors, like your SHP2. The JAK itself is autophosphorylated, so it binds to the SOX3. Here's SOX3 binding down here to these two phosphorylated tyrosine residues. Here's STAT3. These are all signal transduction cascade proteins that I've talked about that themselves are kinases. I've talked about various episodes of VeraMed and Authentic Biochemistry. This turns on the JAK-STAT pathway. JAK-STAT pathway is a very similar, actually identical pathway that you get for the cytokine receptors. So see, adipokines act like cytokines because they're turning on the JAK-STAT pathway. This GRB2 also, when it's phosphorylated and associated with SHP2, will turn on the RAS-RAF. This can be an oncogenic problem sometimes. So let's go back to here. Once you hit the STAT3, that dimerizes and translocates to the nucleus. See, and that can cause transcription. All right, that's what that does. And what kind of transcription? All kinds of genes, including the suppressor of cytokine signaling, the SOX3, which is here controlling the leptin mediator response, right? And that's actually acting as a negative feedback signaling. Additionally, leptin induces the activation of SHP2, that's up here, which then recruits the adapter protein GRB2. I just showed you that. Prompt activation of RAS and RAP. When those are mutated, you get cancer. MAP kinase, okay? All of that then signals. Leptin also mediates PI3 kinase and the AKT pathway. This is over here. It's the IRS 1-2 pathway, P85. There's a P13 kinase phosphorylating AKT. Again, these are canonical pathways that are utilized by multiple signal transduction cascades. Here's the one showing you with leptin, okay? So leptin can function, again, very much like a cytokine, which is why it's called an adipokine, right? So it works, again, with the insulin receptor substrate 1/2. That's what the IRS is. So this is normally turned on by insulin, you see. And there's also a tyrosine phosphatase 1B, or PTP1B, and that acts as a negative regulator of leptin signaling because that's going to remove all those phosphates. So it's also helping to control the tuning down of this induction, okay? And it works through dephosphorylation of the JAK2, which would shut everything down because it prevents this autophosphorylation of all those tyrosine residues. All right. So a clue as to the identity of factors that regulate energy balance was provided by the identification of the recessive mouse obese mutation. That's known as the OB, okay? And a subsequent identification of the DB spontaneous mutation. So when you look at lipid metabolism, you got OB mice and you got DB mice, okay? And those are what helped us understand what leptin does, okay? That's how leptin was discovered. Basically, not that long ago in terms of overall understanding of biochemistry, like in the 90s. All right, so both of those cause mice to develop massive obesity, mutations so the DB mutation, the OB mutation, and they show identical phenotypes on the same genetic background. So it looks like you've got one molecular signaling paradigm there, okay, even though one causes diabetes and one just causes obesity. So similar phenotype also develops in genetically obese rats, and that's the FA mutation. So you've got a rat model, you got a mouse model, okay? In mice, you got OBDB, and rat, you got FA. These fully penetrant, that means they can be genetically transmitted, they go through meiosis, and you can get double recessives, and you can get these mutations, and you can get very obese mice or very diabetic obese mice, or even rats, if they have the FA mutation. So those are recessive mutations. If you get a double mutation, you get a really, really, really obese mouse, very sickly. Causes extreme obesity. It causes insulin-resistant diabetes. I just said diabetes phenomena. As part of a complex syndrome, including a broad set of unusual abnormalities, not generally observed among obese humans. So the mouse model, rat model is not the same as what happens in humans. Now, but in mice, you get infertility, immune alterations, and hypothermia. Mice tend to regulate their temperature not so much by shivering thermogenesis, but by non-shivering thermogenesis, such as the regulation of uncoupling protein, fatty acid binding to that, and then allowing for electrons to leak out of the mitochondria, generating heat that way. Okay? So the extreme obese, because they have a lot of brown fat, rodents do. The extreme obesity of these mutants resembles that of animals with lesions in the ventromedial hypothalamus. So that's interesting. So you have a mutation in the VMH, something wrong with that part of the central nervous system, right? Of that component of the hypothalamus, which regulates several metabolic and signaling phenomena from the CNS. Looks like then you've got these mice that are obese, and you got these mice that are diabetic, and you got the rats that are obese or diabetic, and it looks like it's signaling to the brain. So it looks like you're starting to figure out that this is a component that is signaling to the brain from the adipose, right? All right. Animals with a hypothalamic lesion overproduce a blood-borne signal that normally decreases weight, but because of the lesion, they can't respond. The lesion was further suggested to lead to a compensatory increase in the level of a humoral factor coming in from the blood, thus resulting in weight loss in a paired animal with normal hypothalamus. Okay, this is via doing breeding of these mutants. OB mice are obese because they don't produce a hormone that regulates food intake and body weight. DB mice and FA rats are obese because they do not express the receptor for that factor. And the receptor is expressed, of course, in the hypothalamus, the VMH of the hypothalamus, okay? Now, what do you think that is? Yes, for those of you out there that are screaming leptin, you are right. All right. OB was identified in 94. I told you this relatively recently in many ways. I was well past graduate school by then. Through positional cloning and shown to be expressed in adipose tissue as a secreted 14 kilodalton polypeptide that circulates in plasma in normal animals. Its levels are elevated in DB mice and it decreases after weight loss. Administration of the hormone, they named leptin, okay, was found to significantly decrease body weight and the adipose tissue mass in the OB mutant mice and wild-type animals too, but it has no discernible effect on the DB animals. And you know why? DB animals is a receptor problem, not an adipokine problem, you see? Yep. In contrast to weight loss after dieting, leptin had no effect on decreasing lean mass. So it looks like it's not directly linked just to the normal feed-fast system, which tells you what? Which tells you there has to be an organ that's associated beyond just normal digestion. What would that organ be? Adipose. There you go. Leptin levels increase when fat mass increases. Here's your answer. Thereby suppressing food intake, whereas weight loss leads to a decrease in leptin levels and a consequent appetitive state, increasing in food intake. Your appetite increases. This mechanism maintains a homeostatic control, which is what we're looking for all along, of adipose tissue mass with a relatively narrow range of leptin content, leptin receptor mediated responses, thus serving as an important evolutionary function, obviously, for controlling the oligenous state of the mammal, particularly human. And of course, the rat and mouse models are just models. So leptin acts primarily on a cytokine family receptor encoded by the DB gene. DB gene is for the receptor, expressed in discrete neural populations in the hypothalamus and actually elsewhere in the brain. Okay, so lots of other places. And here we're taking a look at this figure. Let me move me out of the way here, put me over here. All right, so here you got food intake, right? So you got leptin is sending a signal, okay? Leptin is being controlled by adipose. Here are the various neurons that are involved, NPY, AGRP, as well as the POMC neurons. These are specific neurons that are that maintain the leptin receptor, the DB gene product. Okay, so all of this is regulating food intake. That's what it's showing you here. And muscle is going to be, of course, a reservoir for controlling energy homeostasis, because skeletal muscle and even cardio muscle is going to be using energy. So it's got to be linked into the axial couple. So leptin is an afferent signal in a negative feedback loop. That means it shuts down appetite when it builds up, maintains homeostatic control of adipose tissue mass, and links changes in energy stores to a set of adaptive physiological responses. The signaling form of leptin receptor, that's the LEPRB, is expressed primarily in the brain, okay, in the VMH of the hypothalamus. And modes of leptin's effect on end organs are indirect via the central nervous system, then signaling back out, see? Leptin also regulates immune function, as I've been alluding to, via the CNS again, and also acts directly on immune cells. So it works via the CNS, but also directly on immune cells as a signaling molecule. So there are receptors for leptin. It also expresses high levels of leptin RB. That's the receptor that's in these immune cells. Leptin regulates the activity of key neural populations in the arcuate nucleus of the hypothalamus, where it inhibits the orexigenic NPY AGRP. Okay, orexigenic means inducing appetite, while stimulating anorexigenic POMC neurons. Okay, so negative on those neurons that have NPY AGRP, which are going to turn on the appetite, and so it'll be negative on those neurons, positive on the POMC type of neurons in the hypothalamus, and so pro-opiomelanocortin neurons, and that's going to then have a positive effect, and it's going to depress appetite. through those neurons. So leptin also acts on other brain regions besides these. Regulates behavior, metabolism, thermogenesis, as I alluded to, particularly in those with brown fat, like in rodents. The neuroendocrine axis, immune function, and many other physiological processes. So it is a very important compound, very important adipokine. So there are several splice variants, as you might guess, of the leptin receptor. Okay, so this is RNA that's spliced differentially, so you're going to have different isoforms of the receptor. But all but one of the different mutant strains of DB mice and FA rats carry null mutations, and those affect all the splice variants, obviously. However, the original DB strain, maintained by the C57BLKS genetic background, that's in the mouse strain, has an unusual mutation, it's a rare one, affecting only one of the splice variants, and we call that OBRB, while leaving the function of all the other forms unaltered. So that OB we first picked up, when researchers first picked up, was pure propinquity, because that was one that had only one splice variant. Therefore, we're able to isolate that system because of that. The OBRB isoform is a long cytoplasmic region containing multiple motifs. This is about the protein itself, the mutated protein required for signal transduction, whereas the other isoforms don't have all of that regulatory, covalently modifiable regulatory system, because that's the long cytoplasmic region where all those tyrosine residues are. Yep. In addition, whereas the other splice variants are expressed in many tissues, the OBRB is highly enriched in the hypothalamus of VMH. And since the phenotype of this mutant is identical to those of animals with null mutations, because you're not phosphorylating those tyrosine residues, not doing all the signal transduction cascade we just saw, the findings provided initial evidence that leptin acts primarily in the hypothalamus. In agreement with that hypothesis, plasma levels of leptin increase after hypothalamic lesions. Why would leptin increase? Because it's not binding to its receptor. It's increased in synthesis because it's not receiving the signal. The receptor's gone because of the hypothalamic lesion. You see, the receptor is not functional, so leptin keeps on getting jacked up. Low doses of intracerebroventricular leptin, introducing it that way, replicate the effects of much higher doses of peripheral leptin coming in through circulation. So that tells you it's targeting that region, which is the hypothalamus. All right. Now back to looking at a nice diagram here, again from this paper. Leptin's binding to its receptor. There's your tyrosine residues. There's a JAK. There's the ARS. Now it's showing you a few more things. What happens when leptin binds? It shuts down the Amp Kinase, okay? Shutting down the Amp Kinase is an important feature regulating metabolism. Here's the STAT3 phosphorylated SOX3 generated from transcription going through the signal transduction cascade using these factors that are phosphorylated, working under the influence and function of transcriptional factors, right? There are other channels involved. The ionotropic ones, there's a potassium channel, potassium ATP channel, which also seems to be regulating this whole pathway, but it's not clear exactly how. But we know if we do something to modify the leptin receptor, we still get some signaling into this pathway. Likewise, if we inhibit this ionotropic channel or this particular potassium ATP channel, we can modulate the leptin receptor. So let's read over here what all this says. The leptin receptors is, again, a cytokine family receptor. Activates the JAK kinase. Phosphorylation of all those tyrosine residues, turns on STAT3 to that particular tyrosine residue. This residue is then phosphorylated. SHP2 is activated, binds to another tyrosine. And on we go with SOX3, and inhibits the JAK2 and diminishes leptin signaling since it's a feedback regulation from the SOX pathway. That's really important. You got to understand that once that's turned on, it starts turning down, right? It starts titrating down the response because you don't want the response always on. All induction by cytokines have similar sort of built-in rheostatic control to start trimming down the activity of the receptor as soon as it binds its ligand. This particular cytokine receptor, adipokine receptor, is no different. In fact, it also is controlled by this phosphatase. That's a pyruvate-tyrosine phosphatase. Although the factors regulate its activity in leptin-responsive neurons are still not known. That would have been the reference from this paper. Several other signal transduction pathways are activated by leptin, and although inhibitors of these pathways have been shown to blunt some of the leptin's actions, the cellular mechanisms controlling their activation in neurons hasn't been characterized. Leptin signaling in neurons has been reported to regulate neural activity in part, as I told you, this potassium channel, potassium ATP channel, those signal transduction regulating this in the other channels, right here, hasn't been well characterized yet, but people are working on it. Okay. You know that how leptin activates some neurons, such as I told you, the pro-opiomelanocortin neurons, while inhibiting the NPY and the AGRP, that's the Agouti protein, and other neural populations. It still remains unclear why it does this differential regulation of those two. It's not just the receptor, it's obviously something distal to receptor binding to the leptin, right, in those particular neuron beds, right? Amp Kinase and whatnot are all being regulated via this metabolism. And here's all your acronyms, but we've already covered them. All right. So, what about the immune response? This comes from Frontiers in Physiology, published a year ago. You know, I do a lot of immunology in my Vera Med lectures and in my authentic biochemistry. So hopefully you've listened to some of those and you know a lot about innate versus adaptive immunity. I'm not going to cover a review of that right now. I'm just going to jump right in and tell you that leptin regulates both innate and adaptive immunity, something I alluded to in my introduction. So how? Leptin controls granulocytes. It can activate them. It can also cause it decreases apoptosis since it activates. It stimulates cytokine release. What kinds of cytokines? I'll show you soon. And it also helps regulate chemotaxis. That's all in the granulocyte states cells of the innate immunity. Granulocytes, of course, produce hydrogen peroxide. They produce these ICAMs and also CD18, which means the signaling through that plasma membrane protein, and they inhibit ICAM3. So they turn on ICAM1, they inhibit ICAM3, and they inhibit L-select. Now, these are proteins on the surface of cells which regulate cell-cell contact interactions, right? So these are adhesion molecules associated with the matrix, right? Granulocytes. What about M1 macrophages? Well, it activates M1 macrophages. Those have a nitric oxide synthase 2 mediated response. IL-6 pro-inflammatory cytokines, M1 macrophages induce inflammation or are appropriate for the inflammasome and the inflammatory system. So what does leptin do? It works through macrophages by kicking up M1. It can M1 can convert to M2. It does so then ultimately by expressing Arg1 and interleukin 10, something I covered very recently in the Vera Med lecture. Mast cells are involved in this regulation of macrophages. So again, you can get increase in activation, increase in cytokines, increase in phagocytosis, which is what macrophages do, but plus, remember, they're antigen-presenting cells, professional antigen-presenting cells. Leptin also controls natural killer cells, increases the cytotoxicity of them, increases cell proliferation of them, and it decreases their own programmed cell death. Leptin works through dendritic cells. Again, these are really important innate immune cells because they function to talk to the T lymphocytes. It allows for dendritic cells to mature, migrate, usually with an MHC class II bound antigens. They become professional antigen-presenting cells, and it decreases the apoptosis. So see how this can all turn on an inflammatory response. In terms of the adaptive immunity, so this is all the innate immunity, which would feed into the adaptive, but directly leptin controls T cell maturation, as I said. Th1 giving you interferon gamma and IL-2. Those are really potent pro-inflammatory cytokines. Th2 responses like interleukin 4. Sometimes it's differential, as I mentioned, between Th1 and Th2, at least in the mouse model, not so much in humans. But all of this induces proliferation and responsiveness of these pro-inflammatory T cells. Also, Th17, something I also just covered in authentic biochemistry and in my Vera Med lectures. Th17 often is involved with autoimmune responses. It's also what's triggered when you get a T cell response from a vaccine. that produce interleukin 17. That's why they're called Th17 cells. Interleukin 17 is pretty important pro-inflammatory cytokine. But also T cells can be differentiated into Tregs. Leptin tends to decrease that pathway. So when you decrease the amount of Tregs, what happens? You decrease the ability to suppress the T cell mediated pro-inflammatory response by these three classes. Okay. All right. Ultimately, too, leptin works directly with B cells, right? Turning into plasma cells eventually. It can increase their proliferation. It can decrease their apoptosis, which means they're active longer, they're alive longer. Increase the amount of cytokines that B cells produce, and also the development of the B cells fully differentiating into plasma cells, and therefore regulating what? Right? The synthesis of IgG, right? So think about all the immunoglobulin classes that are being turned on by leptin as well. It's a really important component there. All right. So what about now going back to the actual, now those were from other papers, those are from other journals, that's what I was talking about there. Now we're going back to this review article. Adipose signaling and leptin sensu stricto per se. Leptin modulates dietary intake. That's how it was first discovered, as we mentioned, working through those POMC neurons in the VMH, right? And body mass via stimulating the pro-opiomelanocortin, the POMC neurons, inhibiting the neuropeptide Y and the Agouti-related protein neurons. Already mentioned this. That's all happening in the arcuate nucleus of the hypothalamus, okay, which is proximal to the median eminence, and that is a circumventricular tissue providing a porous blood-brain barrier. So this allows for the blood-brain barrier to open up to cytokines and to T lymphocytes, you see. So this is a very profound effect that leptin has. So the leptin receptor knockout in the POMC neurons show that these neurons diminish food intake. Normally, that's what they do. And body weight after leptin treatment. So you get rid of that, you get obesity, right? Leptin receptor knockouts like the DB. A CRISPR knockout of the leptin receptor in the AGRP neuron correlated with obesity as well. So there are more than one neural nucleus that responds to leptin. We just don't quite understand what's going on with the Agouti and the NPY system, but we know that those are normally tuned down, but when we get rid of it, we get pleiotropic effects, which doesn't surprise me at all. This is how hormones work, right? Multiple feedback loops, of course. Mutations that disrupt melanocortin signaling in animals and humans cause leptin resistance and obesity, thus suggesting that they also mediate some of the leptin effects, of course, okay? Because melanocortin is a component of what's turned on by the POMC. POMC neurons then also regulate glucose metabolism in response to leptin independently entirely of body mass, okay? So this is all associated with the type 2 diabetes we sometimes see. So let's talk a little bit more about this arcuate nucleus, and then we're going to stop here for today and finish this entire PowerPoint and put it into number 2, because I don't want you to be fatigued by such a long variable lecture. So what do we have? We have activation of Amp Kinase. When that happens, it increases appetite. This obviously is the reverse effect from leptin, right? So that's why Amp Kinase is tuned down by leptin in the POMC neurons of the arcuate nucleus of the hypothalamus. So Amp Kinase is highly expressed in that nucleus, and indeed the dorsomedial DMH, paraventricular PFH, and the ventromedial VMH nuclei, all of the hypothalamus. So those are all part of the lateral, these are all various components of the hypothalamus, but even the lateral hypothalamic area, the LAHA, is regulated via Amp Kinase, okay? So overall, hypothalamic Amp Kinase, but that turns on things like beta oxidation of fatty acids, okay, is part of an adaptive change in the physiological regulation of the feeding cycle. Fasting increases, but refeeding inhibits the Amp Kinase activity. So it makes sense that leptin is working along that same kind of axial corridor in the hypothalamic region. Okay. Now, hypothalamic Amp Kinase dominant negative isoforms. So these are going to be variants in that gene being expressed that gives you a dominant negative effect. They actually decrease the expression of orexigenic neuropeptides, such as Agouti-related peptide and neuropeptide Y. Those are orexigenic neuropeptides in the arcuate nucleus. So that makes perfect sense too. When you have a dominant negative, the AMP kinase no longer signaling through these pathways. Remember, leptin is being negatively correlated to those two domains, the AGRP and the neuropeptide Y, because you're going to be shutting down AMP kinase. So this all fits into our understanding of the signal transduction cascade in the central nervous system. Overexpression of AMP kinase constitutively active, that's a different mutation, elevates the fasting-induced expression of these neurons, as well as expression of the melanin-concentrating hormone in the lateral hypothalamus area. Okay, so you get the idea that this is being regulated by multiple receptor domains, particular subsets of nuclei in different regions of the hypothalamus as they are mediating and responding to leptin, and then down signaling through AMP kinase. So AMP kinase modulates the expression of NPY and indeed even pro-opiomelanocortin by regulating autophagy. So you see the modulation even of the POMC in those POMC neurons in the hypothalamus are also regulated by AMP kinase. There's a negative feedback loop there as well, okay, which again makes sense. All right, so we're going to finish today with just this last slide. Again, we're talking about this region of the central nervous system. There's the hypothalamus. There's the hypothalamus, what it looks like. There's the LHA, the PVH. Remember, this is a bilateral system. So you have the left side and the right side. Okay. This is where you understand the subnuclear anatomical differentiation of the hypothalamus, right? And there's very specific regions here. There's the anti-obesity drug we would want it to bind right here at the AMP kinase locus. And that's where the AGRP and NPY is. And this is in the 3V region leading up into this by-system anatomical controlling area of the hypothalamus, right? And this is indeed where adiponectin, AGRP, glyph, glucagon, glucose itself, and various other hormones are regulating right in this region in the VMH. So AMP kinase senses several nutritional hormonal stimuli. The activity of hypothalamic AMP kinase, that's adenosine monophosphate kinase, is mediated by specific regulation of the sympathetic, the CNS, and the parasympathetic, what's controlling the whole system in the reverse, or the PSNS. The fact that inhibition of the hypothalamic AMP kinase leads to anorexia, okay, that would make sense, right? And increased thermogenesis and therefore elevated energy expenditure makes it an interesting target for drug development. That's what this paper was telling you. With its potential for controlling both the sides of the energy balance equation, there's a 3V region down there. That's the third ventricle. So you have the AGRP, Agouti-related peptide. You have the arcuate nucleus of the hypothalamus, the BMP89, that's the bone morphogenic protein. These are all found down here. Remember, the GLP is glucagon-like peptide 1, and these are the other, the PVH, you already know what that is, neuropeptide Y. You also have thyroxine, which is 3'5 triiodothyronine binds. VMH is, of course, the ventral medium nucleus of the hypothalamus. So all of this region of the hypothalamus is associated with body weight and energy homeostasis. It's associated with turning white fat into brown fat, which will also allow for more beta oxidation. Brown simply means there's more mitochondria, more carnitine palmitoyl transferase, more beta oxidation, more NADH and FADH production to run through the TCA cycle so that you metabolize more fat. More brown adipose, more metabolism of fatty acid. Okay, so you also get glucose sensitization, and you get lipid oxidation in the muscle. All of this is good stuff, right? Thermogenesis in the brown fat region. This would be in the rodent model. Food intake is controlled by this region, as we've been saying. And the overall glucose and lipid metabolism is regulated in the liver, probably via also signaling back from this region of the hypothalamus. So we're going to stop here. Let me find my region for stopping here. End the show here. Close off this PowerPoint. We're going to come back to this. So you can see that we've gone through about 16 slides. We're on slide 16. There's about 10 more to go, right? And I'm going to follow this up with part 2 of our talk on leptin. And we're going to get much more into the signal transduction cascades intracellularly. And then we're going to blow it back up and talk a lot about physiology and how hyperleptinemia and hypoleptinemia are associated with a host of very important pathophysiological states in human disease, right? So that's the next part of it. And please stay tuned. I'm going to try to publish that sometime very soon, the next few days, so we can get this leptin story out there. This is all background information. And then we're going to start probing the primary literature for papers just recently published on leptin. And the first thing I'm probably going to want to do is go into the immune response, because it's a very hot topic and one I'm particularly interested in, also clients are. So for right now, I'm going to say this is Dr. Daniel Guerra, and bye for now.